Tá, vamos só dar um segundo aí para...
Ele está fazendo o setting aqui, tá vindo devagarzinho. Daqui a pouco vai abrir aí. Já abriu a janela para vocês? Não.
Not yet.
Ainda não aqui também.
Para mim, não.
Pronto, começando a abrir.
Live on YouTube.
Então,
Okay, let's see.
beleza. Manda bala.
Are we live?
Yes.
Okay. All right, so, you know, we have this weird tradition in the cell death field that when we introduce a very close friend, we talk, we say a very funny and embarrassing joke and or a situation, a story about the next speaker, to break the ice and, you know, to make fun of the person and show that she's a human like us and she'll be accessible. And I have a lot of stories about Larissa, the next speaker, but I won't say a fun story about her, because as you will know through her talk, she's very approachable, very nice, very fun person. So I'll give her a pass. Larissa graduated at the University of São Paulo under the supervision of Dario Zamboni. And then she moved for a postdoc in Dr. Doug Green's lab in Memphis, Tennessee, St. Jude Children's Research Hospital, where I have the honor to know her much better and be a witness of her work ethic and how impressive is her line of thought and her line of experimentation. So I won't go much further in the presentation, just saying that she's currently a young investigator from FAPESP, and she holds a tenure track position at the University of São Paulo, Ribeirão Preto. She has published very, very cool papers, and I won't talk about it because she will. So Larissa, thank you very, very much for being here with us and presenting your work and your views on this amazing process. So please feel free to take the lead and let us know what do you prefer, if we hold the questions to the end, or if it's better to interrupt you, or if you want to give some breaks and then we can make blocks of questions.
Feel free to interrupt, Ricardo, whenever people have questions. I mean, if you prefer to collect some questions and stop the same way you did with Peter, I think it's a great idea. Well, sorry. Thank you. Thank you a lot, Ricardo, Gustavo, Karina, for the invitation. I felt really honored because it's a very international meeting, clearly. And it's an honor to be the Brazilian representative. I hope I live up to the stellar team that we put together. So, are you seeing my? You're not seeing my screen yet, right? I think Gustavo needs to.
Larissa.
It's okay. Should I wait a second?
Wait a second. I think I already granted to share screen. Yes, you should be.
It says host disabled attendee screen sharing. No, you haven't authorized me yet.
Hey, come on! I authorized everyone
except me.
Well, what I'm gonna do is I'm just gonna make you co-host.
The Brazilian bullying.
I make you co-host now, so you should be able to do that.
Oh, share screen. Oh, okay. Yeah.
There you go.
All right.
Right.
So let me. Okay. So where is the pen? Okay. So as Ricardo introduced me, I'm actually a young faculty member here in Ribeirão Preto. I had my PhD here, came back home, And I've been following up in the research I performed during my postdoctoral training with Dr. Green in Memphis, and I'll show a little bit of that. But I think this is not a regular seminar. This is a teaching class, so it's not going to be that much about my work. The same way that Peter, Christina, and Seamus gave their amazing talks before me. So this is going to be on the consequences of recognition of dying cells, specifically by innate immune cells, and mostly what I'm going to talk about refers to macrophages. So keep that in mind. I'm emphasizing that because efferocytosis field is actually booming right now. So there are a lot of literature, a lot of interesting literature, and it would be possible to put everything together in a logical and, in a way that you could absorb anything between 10:30 and 12 right before lunch. So feel free to ask any questions if I'm not talking about the subject that you're interested, or you can write me an email later. I'll be happy to try to help. So what I'm going to talk about today is actually centered in 3 main points. The first part is going to be focused on the mechanisms of sensing, internalizing, recognizing and internalizing, and degrading dying cells. And here I say dying cells because most of the knowledge we have has been built in studying responses to apoptotic cells, but we are not sure by now if this is only the case. The second point is going to be focused on the effector mechanisms of the innate immune response in immune cells. So as I mentioned, mainly macrophages in response to efferocytosis. So what happens once this corpse has been internalized? And the third point, which is going to be the one more broad, I'll talk a little bit about the determinants of the immune response to efferocytosis. So the most well-characterized response is by far recognition and engulfment of apoptotic sterile cells. And I think there is a lot of knowledge being built right now showing that it's way more complicated than that. So I'll talk a little bit about that. So if this is an immunology course, I think that this picture is no novelty for most of you, right? So this is the way that we depict the inflammatory response during an infection. So if you have an insult that it's caused by the invasion of a pathogen, and then we can extrapolate that for situations when you have a sterile insult, but here I'm going to talk about the pathogen. You're going to have at least 2 well-defined phases. One that it's heavily associated with the control of these invading organism, and a second phase that is concomitant. It's a little bit later, but it's concomitant with clearance, that it's marked by retraction of the inflammatory responses. So here we have extremely regulated pathways that are going to fulfill at least 2 main mechanisms that are going to make it possible to survive over the infection. One is to trigger anti-inflammatory responses that are going to reduce the inflammatory milieu, control the secretion of inflammatory cytokines, stimulate the secretion of anti-inflammatory cytokines and resolvins, pro-resolution molecules in general. The other very important function is actually to deal with the damage of the infection and of the inflammatory response. So you're going to have damage caused both by pathogenic action of the invading organism, but you're also going to have a lot of damage caused by the immune response itself, either by the effector mechanisms or by simply by the migration of immune cells to the site of infection and their death after they play their role. So if you don't have control of inflammatory response, and if you don't have efficient repair of damage, either of them can actually cause a very problematic scenario that it's associated with chronic inflammation and immune paralysis. So you have excessive inflammation, and at the same time you can identify markers of pro-resolution program being developed, but it's not effective, right? So it's a mixed response. And you have several pathological scenarios that are going to come of these defects on the resolution phase of inflammation. And there is something else that I want to point here. We have this picture of these phases of the inflammatory response very well delimited, but restriction of the damage is actually occurring together with the clearance of the pathogen. So the battlefield needs to be controlled, otherwise you might have death, or you might have excessive morbidity, simply caused because the damage is too much. There is no physiological balance, or at least physiological minimal homeostatic control that allows the mechanisms of restriction of pathogen growth to occur. So this has this concept of the balance between resistance to pathogen, the mechanisms that act by eliminating the invading organism, and the mechanisms that provide tolerance to the disease. Both of them occur during the phase of clearance of the pathogen, and you have situations where not necessarily you're going to have full control of the pathogen, but if you have control of the damage, that might be enough to at least postpone failure and give time to the organism to establish minimal homeostasis to deal with the pathogen later. Okay, so this is actually very important, and might be able to explain several infectious scenarios when you have when the host succumb to infection. And restriction, so pro-host defense responses, either during the resolution phase or either promoting tolerance to the disease, you're going to have different cells that participate in the orchestration of these responses, or by actually acting like effector cells. But we definitely have a major role for professional phagocytes, especially macrophages, in these responses. So I'm sure it is a novelty for anyone here in this audience, but we have this already absorbed definition of macrophages as cells capable of polarizing their function according to the stimuli. Okay, so we have defined these extreme scenarios when a macrophage behaves like an extremely inflammatory cell that is going to contribute for mainly for pathogen clearance and activation of inflammatory mechanisms. And you have in the other extreme macrophages that, depending on the stimuli, they become extreme anti-inflammatory pro-resolving cells. The fact is, this dichotomy, as we define, not necessarily apply to physiology, even though you can always most of the scenarios you can identify some level of commitment of the macrophages to one or the other kind of response. So it's a complicated model, but it still helps us to understand a lot about macrophage physiology and immune responses. So now we accept that depending on a combination of different signals, and here we can call them randomly A, B, C, D, the macrophage will either program their genetic expression toward pathogen clearance, and usually this is triggered by recognition of molecular patterns associated with the pathogen in combination with other stimuli such as inflammatory cytokines coming from Th1 response. And that will drive genetic programming that will culminate in the production of mediators and cytokines that are important for pathogen clearance, either directly by the macrophage or by recruiting and activating other cells of the immune system. On the other hand, you have stimuli, especially those that are related to Th2 dependent responses that are going to drive this anti-inflammatory pro-resolution phenotype. And these signals, for a while, they have been treated as promiscuous. Once you have IL-4 or IL-13 or apoptotic cells, you're going to have commitment with a full anti-inflammatory pro-resolution programming. This is not true at all, and it's becoming clearer and clearer. And I'm going to go back exactly to this example of the combination of Th2 cytokines plus recognition and engulfment of apoptotic cells. And Seamus talked about this yesterday, and Christina as well. And we know that this clearance of dying cells, resolving inflammation, either resolving or causing immune suppression, it's not something that happens only during insult, during infection. This is actually fundamental for homeostasis. And as mentioned yesterday, and I'm just going to reinforce here, we have a huge amount of cells dying any single moment in our body, to the extent that in an adult male, in 10 days, 1% of his cells will be will die and be cleared. And this is extremely important because it is clear by now that cell waste, it's a major source of molecular patterns associated with danger. And DAMPs, they are the signature that alert the organism that you're having cell death in a dangerous way. So making sure that in a homeostatic situation, the garbage is cleared to avoid the release of DAMPs promiscuously, it's a fundamental requirement for multicellular organisms. And this clearance of dying cells, it's a complex process, but we have identified some steps of the way that are better understood. So the process of sensing, engulfing, and degrading cell corpses, this triad we call efferocytosis, which means by definition to bury a corpse, to get rid of a corpse. And you have macrophages and other professional phagocytes, they can recognize dying cells because these cells in the process of dying, they are going to release signals that professional phagocytes can sense. So we call them find me signals, and you have several of them that that are important. It doesn't seem to have one that it's pivotal because a lot of work made in knockout mice has shown that one can substitute the other. This is a little bit tricky. But once a macrophage identifies a dying cell that releases find me signals, you're going to have either direct or indirect interaction between the cell corpse and the professional phagocytes. And this happens through signals that we call eat me signals. So the cell membrane of a dying cell exposes molecules that are going to be recognized by a professional phagocyte. And definitely the most important one, and one that was mentioned yesterday and today, is the presence of phosphatidylserine in the outer membrane. So this phospholipid is kept on the inner membrane of living cells through flipases, and once you have activation of dying pathways, in the case of apoptosis, we know how this happens. You have activation of caspase 3, and caspase 3 is going to cleave lipase. Then you lose this mechanism of flipping, and now you can have the presence of phosphatidylserine in the outer membrane. And most of the receptors that recognize dying cells, they act by recognizing either directly or indirectly the presence of phosphatidylserine in the dying cell. But it's not the only one. And then besides that, you're going to have, once the corpse is internalized, you're going to have activation of degradation or degrading, right? Always confused, degradation machinery. And this part here is actually one that, even though it's clear that macrophages, professional phagocytes, they are going to engulf corpses, this knowledge has been built in more than 1 century. I think that only in the last 20 years, we've actually started to understand what it means to eliminate a cell corpse. And I'm going to talk more about that later on. So as I mentioned, we have several receptors that can recognize a dying cell, and most of them are going to recognize phosphatidylserine. And this seems to be a promiscuous relationship. To the best of my knowledge, you don't have one single sensor that it's preeminent. All of them seem to be important. I can highlight to you the ones that we've built more literature on, that are the members of the tyrosine kinase, the TAM family of tyrosine kinase, where you have MER, AXL, and TYR3. The TIM receptors, TIM 1, 3, 4, but the only one that it's strictly related to efferocytosis in an optimal way is TIM 4. You have integrins that also are important for the recognition indirectly of phosphatidylserine, and there is one that it's not in this picture that are scavenger receptors. They can also bind phosphatidylserine, not only phosphatidylserine, but this is one of the phospholipids that they can recognize, and this can lead to efferocytosis. But as I mentioned, it doesn't seem to have one that it's absolutely necessary, but I'll go back to that later. But we have genetic evidence in mice models that whenever you get rid of one of these receptors, you do have some sort of chronic response being triggered. And this process of sense, bind, internalize, and degrade a dying cell that we call efferocytosis, as I mentioned, has been understood in terms of recognition of apoptotic dying cells. And because of that, our best understanding is that efferocytosis drives suppression of immune responses. So recognition of apoptotic cells in general by macrophages it's considered telerogenic cell death, and contributes to this balance, to the skewing of this balance from an inflammatory to an anti-inflammatory cell type. This anti-inflammatory cell type that can be either immune silent or that can actively activate anti-inflammatory pathways, pro-tissue repair pathways. And because efferocytosis has a well-recognized role in cleaning apoptotic cells and preventing inflammatory responses, being very immune suppressive, there are several pathological implications that have been connected to a failure in this process. So we have several autoimmune disorders and autoinflammatory disorders that, to some extent, mostly from mice work, could be connected to a defect either in sensing, binding, or degrading a dying cell. Okay. On the other hand, we also know that this immune suppression, this anti-inflammatory response triggered by the engulfment of apoptotic cells can also be pathogenic. And the best example, in my view, is in the cancer scenario. So immune suppression in the tumor microenvironment can be driven by the recognition of dying cells in the tumor microenvironment. The expectation is that mostly apoptotic dying cells, and that drives several immune suppressive mechanisms. And in the context of tumors, this apoptotic tolerogenic cell death has been used as an example of the difference between immune silent tolerogenic cell death versus an immunogenic cell death. So I'm not going to talk today about these contrasting functions. You're going to have a whole class on that. I just want to emphasize that in the tumor microenvironment, you're going to have the situation when professional phagocytes, not only macrophages, but tumor-associated macrophages for sure, they can sense, find me signals, they can bind, eat me signals, and this is going to contribute for the suppression of innate immune responses. The consequence of the suppression of these innate immune responses, that's going to happen both in DCs, macrophages, neutrophils, is the lack of activation of optimal adaptive immune cells. You're going to have suppression of CD8 positive T cell cytotoxic activity. You're going to have stimulation of the activity of the Tregs, and this whole scenario will lead or contribute to tolerance towards the tumor. Immune suppression in the microenvironment that actually allows the tumor cells to thrive. Okay. Okay, but then we have this scenario where a professional phagocyte such as the macrophage can sense a dying cell, bind, internalize, and degrade this corpse. This is going to generate. This is going to be immune suppressive or anti-inflammatory or stimulate tissue repair programs. But the question is how. So the very first, maybe the most obvious reason, and that it's connected exactly to this idea of tolerogenic versus immunogenic cell death, is that once you engulf a dying cell early on, before it becomes late apoptotic and a necrotic further on, you're going to eliminate potentially dangerously damps from the extracellular environment. This is quite obvious, right? The other reason is that these professional phagocytes, once they engulf dying cells, they are also very efficient to eliminate potentially intracellular dumps that can be generated if the cell engulfs a corpse but doesn't efficiently eliminate this corpse. And this seems actually pretty straightforward, but it took a while to actually start being studied in depth. I think this is a better way to put it. And then obviously you're going to have activation of anti-inflammatory signaling pathways. You're going to have active suppression of inflammatory signaling pathways. And you're going to have stimulation of programs that allow the continual removal of dying cells. And this is fundamental for efficient injury repair. All I want to say here is that it's not that the corpse is internalized, period. Is that the corpse is going to be internalized, and then you're going to have several signaling pathways inside, from these professional phagocytes that are actually going to generate this immune suppressive anti-inflammatory response. So this is an active process. And among the pathways that are going to be modulated, you're going to find a very broad literature. My personal take is that we see a lot of situations where stimulation of macrophages with dying cells is going to obviously visibly modulate one pathway of the macrophage, but I think we still know very little about how. What is we have like a general notion of what's going to be more likely, but proof of concept is actually lacking for the activation of most of these pathways. So I just put this in one slide because the information here is actually, it makes sense because these are pathways that are either extremely inflammatory or either extremely anti-inflammatory. And if efferocytosis can modulate inflammation in any direction, it's going to be quite direct that they are going to be modulated in response to efferocytosis. So efferocytosis can repress NF-κB activation, can repress STAT1 signaling, can induce IL-10 and TGF-beta transcription by regulating the activation of different transcription factors that can act in the promoter region of these cytokines. It's going to promote the synthesis of pro-resolvins. It's going to upregulate the expression of efferocytic receptors, which means it's going to help, it's going to increase the capacity of these professional phagocytes to continually engulf dying cells. And it's going to regulate the processing of metabolic cargo. So there is no novelty on this. I think that the big point and that I want to emphasize is we know very little how these pathways are actually activated. And one of the ways that we think that an engulfed and processed dying cell can modulate signaling pathways of the macrophage, of the professional phagocyte, it's by actually releasing lipid mediators, molecules, because of the digestion of this corpse that can act as signaling molecules. So macrophages, they express nuclear receptors that recognize binds directly to lipids like fat acids and oxysterols. So PPAR gamma, it's a binder of fat acids. LXR, it's a binder of oxysterol. So it's quite obvious to think that once you have a cell corpse that has been degraded, you're going to generate lipid mediators that are going to be released from the phagolysosome. And now once they are in the cytosol, they can signal. This hypothesis has never been directly proved, but knockouts for all these receptors, PPAR gamma, LXR, all of them develop some kind of autoimmune syndrome as they age, and spontaneous autoimmune syndrome. So a lot of information here. The only thing you need to pay attention is this pattern. All we are seeing here are autoantibodies, either against DNA or against nuclear proteins. So we always see that the knockout for these nuclear receptors somehow these mice, they have higher titers of autoantibodies, okay? But again, and I'll just take a minute to digress a little bit here, so bear with me. It's almost impressive how little we know about the mechanisms of immune activation in response to dying cells, considering that phagocytosis is a century-old known process, and recognizing that professional phagocytes can engulf dying cells is actually right there, right at the beginning, right in the first experiments by Mechnikov, okay? So the first description of phagocytosis actually relates to the recognition of dying cells. And I have a feeling that not only in the case of dying cells, but in phagocytosis in general, we kind of accepted that once a professional phagocyte senses and recognizes a particle and engulfs this particle, it's going to magically eliminate it. And that's why the cells are important, because their effector function is to disappear with the cargo, either a dying cell or a pathogen. And it was only on the last 20 years, as I mentioned before, that we started to gather knowledge in the mechanisms and the understanding that this is an extremely regulated process. And it all changed actually when pattern recognition receptors were first identified and described. So early on, it was clear that they had like a major role in regulating major pathways for cytokine production. And this is just one example, like NF-κB activation for the production of inflammatory cytokines such as IL-6, IL-12. But then early on, it also became clear that activation of these receptors was important to trigger phagocytosis and degradation of pathogens. So without active recognition mediated by a specific receptor, you had easily identifiable defect in this response. And this was actually the first evidence that phagocytosis is phagocytosis in immune cells is a regulated process, and that occurs independently of this modulation of inflammatory pathways. And I think this is an important point. But not only that, I think that the game changer here was the recognition that actually the phagosome is a signaling hub. So if the macrophages in immune cells in general, the topology of the signal, if it's coming from the membrane or if it's coming from the cytosol, is meaningful. It triggered completely different sets of responses, and the lecture from Peter Gross was exactly about that. We know that membrane receptors, they trigger different response from cytosolic receptors. But signals that are coming from the phagosome, they are going to be different from the signals coming from the membrane. And this is like the classic demonstration, very elegant work showing that TLR4 activation in the membrane favors NF-κB activation, but TLR4 sensing from the phagosome triggers preferentially type 1 interferon production, okay? So this is proof of concept that the phagosome is an autonomous entity from these professional phagocytes, and this matters in terms of responses. And this is just reinforcing this concept. And here I'm gonna jump a little bit to later on connect both processes, but one of the cell events that now we know they are very tightly connected with how the phagosomes are signaling to the cell is components of the autophagy machinery. So this is a very, this is just a simple cartoon just to remind you, if you haven't heard about autophagy in a while, what this process is. So autophagy, canonical autophagy, and I might repeat this term again later on, is a process that mediates recognition of intracellular cargo, mainly in defective organelles. And this is important to address these defective organelles for degradation by the lysosome in a compartment called autophagolysosome. And this is obviously important to guarantee control of organelle quality, and this is fundamental for metabolic homeostasis. But it has also been selected as a form of self-defense throughout evolution. So the same way that a defective mitochondria is recognized by the autophagy machinery, you can also have recognition of a bacteria in the cytosol. This bacteria is going to be enclosed in a compartment in a similar way and directed for the lysosome. But components of the autophagy machinery, they can be directly recruited to the phagosome in a situation that has absolutely nothing to do with canonical autophagy. This autophagy that I mentioned before related to stress response, and that deals with damaged organelles. And in fact, these components of the autophagy machinery, they have so many other non-canonical functions that I think that autophagy is actually just one part of the repertoire of functions. They are not necessarily the main one, I think, depending on the situation. But just to illustrate my point, this is actually what we call canonical autophagy in the cell. So this is classic experiment where you have a macrophage expressing a fluorescent probe for autophagy. So it's actually a fusion with LC3, which is a terminal effector of the autophagy pathway. And if these cells are submitted to a nutritional stress, then you're going to have the formation of these autophagy puncta, which represent actually this compartment for cargo degradation. So this is pretty basic, but just to remind you. But then if we take a macrophage expressing these fluorescent molecules, and instead of putting them under nutritional stress, we actually feed them a pathogen or a mimic of a pathogen, pay attention to what happens now. Oh. So it's going to happen pretty fast. You're going to see like a blob in the cytoplasm and the GFP being completely recruited to the compartment. So these experiments, they were performed actually trying to understand the dynamics between autophagy and toll-like signaling. But what was happening here was completely different from response to this metabolic stress. Instead of you having formation of autophagosomes in parts of the cytosol, you're actually seeing at least one component of the autophagy machinery being recruited directly to the membrane of the phagosome. But the message that I want to leave here is that now we know that this process of the signaling that comes from the phagosome, it actually requires components of the autophagy machinery, not all of them, and this is what discriminates from regular autophagy, and this is going to mediate signaling, either to a pathogen and to what we are talking about today, engulfed dying cell. We also have like components that are strict to this process of recruitment of the autophagy machinery to the phagosome, and one of these molecules, and I'm going to talk about it also in the next slide, it's called Rubicon. So Rubicon, the same way it is required for the recruitment of the autophagy machinery to the phagosome. So here you're seeing zymosan, but you can think about this like a dying cells, once is engulfed by a professional phagocyte such as a macrophage, the recruitment of the autophagy machinery will require Rubicon. If these cells are deficient in Rubicon, you don't see this recruitment. And this process to discriminate from canonical autophagy, we call LC3-associated phagocytosis, or LEP. And this is a busy slide, and I promise this is the only slide that I'll touch a little bit on molecular mechanisms. But this is just to emphasize that what we are seeing here it's not the same process that canonical autophagy. So the components of autophagy that are required to sense stress, metabolic stress, do not participate. But once you have a signal in the plasma membrane, and among the signals, you have a dying cell, the moment that this particle starts to be phagocytosed, you're going to have recruitment of components of the autophagy that are going to mediate lipidation directly to the single membrane of the phagosome compartment. And this is fundamental to controlled kinetics of the phagosome maturation and the consequences of engulfing a particle through some of the receptors of the immune response, at least. And emphasizing that this happens in response to pathogens, and this happens in response to engulfment of a dying cell through one of those receptors that bind phosphatidylserine in the membrane of an apoptotic cell, for instance. And going back to what I was talking about, we know how we have an idea how sensing happens. We have an idea how recognition happens, and this is what tells us what's happening in terms of degradation of this engulfed body. So once a dying cell is engulfed by a professional phagocyte, the recruitment of this LEP machinery, it's going to be important to both induce this anti-inflammatory cytokine production and to repress inflammatory cytokine production. Okay, so if you take a cell that is deficient in Rubicon, this component that I mentioned, then you see that in response to efferocytosis, you don't have this production of IL-10 that it's seen in wild-type cells. But on the other hand, you start to see production of inflammatory cytokines such as IL-6. So the message here is the machinery that provides optimal engulfment and processing of a dying cell, of at least an apoptotic dying cell, is important to control signaling. And this is just an experimental proof of concept of the physiological consequences of this process. So if you inject dying cells in a mice, these mice is gonna eventually, these cells are gonna be cleared, and this is, at least experimentally, an immune silence process. But if the mice are defect in LEP, if they are Rubicon deficient, then after several weeks of injection of dying cells, you start to see production of autoantibodies. Okay, so this regulation of the phagosome maturation, once you engulf a dying cell is important to induce anti-inflammatory responses, suppress inflammatory responses, and the effects in this pathway are related, can cause autoimmune, or at least the onset of immune disorders. But we still don't understand how. So this is just like the beginning. So if you have recruitment of LEP machinery to the phagosome, you're gonna have modulation of the signaling pathways. Most likely those signaling pathways that I mentioned a few slides before. But what is occurring between LEP once it occurs, once the whole pathway occurs, and modulation or regulation of gene expression programs, we have no clue whatsoever. I think there is one thing that we should keep in mind that might be, that is actually an interesting possibility, is that it's not necessarily related to how the cargo is processed, but maybe you have, the cell can actually sense optimal efferocytosis and optimal phagolysosomal activity, and these might activate molecular pathways inside from the cell, and it's not going to be a signal from a direct signal from the cell, just internal pathways being activated. And with that, I conclude the first part. Are there questions? Should we stop or just?
Sure, we have a couple of questions. Karina, can you make the first question, please?
Yes. So the question is from April Darcy from Cambridge. Does phosphatidylserine operate as an eat me signal for the scavenging of necrotic cell debris also? Or which factors control the cleanup of cell debris resulting from lytic cell death modalities?
Yeah, that's the part that I was going to escape. I was going to mention a little bit later. So phosphatidylserine is the major eat me signal. And as we saw before by Peter, he showed like amazing videos, and I was going to show some snapshots from the same process. Periapoptotic and necroptotic and cell death are also characterized by phosphatidylserine. So the motive to be engulfed is there, right? So at least in vitro, these cells are efficiently engulfed through phosphatidylserine recognition. So you can take a macrophage, feed with cells killed by these specific kinds of cell death, and they are going to be efficiently engulfed, and at least by genetic ablation of some of these eat me receptors, you can see that they stop engulfing. So it's a signal for engulfment of an apoptotic cell, but there is no reason not to be a signal for the engulfment of lytic cells that express phosphatidylserine in the outer membrane during the dying process.
But how about the cellular debris? Because we can see this phosphatidylserine in the internal membrane. So cellular debris could be removed.
Oh, so they, you mean they can be removed through a pathway independent of phosphatidylserine, right?
No, because phosphatidylserine is expressed in the internal membrane. So in the cellular debris, you can reach this phosphatidylserine in this internal layer of the membrane. So
yeah, so it's complete. So cell debris are efficiently clean as well, and this can be mediated by this recognition of phosphatidylserine, but not only. I think that the other eat me signals might be important as well.
But it's on the same topic. There was a question, and now I think it's appeared in a different way here.
Mm-hmm.
But I think the main topic here is that when you have like a pathogen or infection, you have a lot of signals for the macrophage to go there and find the cells and eat them, right? What happens when you have this more physiological cell death that is widespread, as you said, like billions of cells? How the macrophages find these cells and eat them?
That's a great question. And I think that one of the reasons that eliminating these eat me signals, you still have efficient sensing and engulfment, it's maybe the signal that it's most important in homeostatic situation, it's not well defined, right? That might be the clue. So I think that's the best explanation. I don't think we actually know what's the best sensing signal in homeostatic clearance of dying cells.
Yeah, can I just add something, and then
Yes.
we're questioning in a different layer. First of all, I think in the tissue, when you have apoptotic cell death and you have a breakdown of the cells to very tiny apoptotic bodies, you don't need only macrophages or professional phagocytes. All the other stroma cells can capture.
This has been shown, right? Both in liver model and lung model, that epithelial cells, they can contribute a lot to the clearance.
I don't think in a physiological apoptotic, setup where you have like a tissue turnover, whatever, even if we are talking about billions of cell death, I think the stroma cells can deal a lot with the death. But coming back a bit on the necrotic or the some knockout cells, some of the phenotype that you mentioned, Larry, reminds me of a phenotype of DFF knockouts, or so knockouts that are not able to fully digest the DNA.
Yes.
So although you have phagocytosis, engulfment, and recognition of any of these eat me signals, the cells that take up the dying cells cannot really deal with the whole genome, the DNA, and then those animals will develop autoimmune diseases. And so how can you comment a bit on this?
Yeah, I can show you data on that in a bit. What about that?
Okay.
So we'll wait for that. And maybe another question that we might wait for it as well, but I'll put it right away because I don't know if you're going to touch on that subject as well, is that you were talking about like digestion of apoptotic cells and infection, but we know that LAP is also related to cancer, right? So a question here is, any thoughts on Phagosome target drugs on cancer therapy? If you can talk
this is gonna go on what I was discussing with Gustavo, but I'm gonna mention this now so that you can keep that in mind while I'm talking. I think that drugs that target the phagosome, the process of digestion are not in the clinic for a reason. And I think the reason is because you're messing up with several physiological pathways that mess up with the activity of the immune cells, parenchymal cells that are participating in the tumor microenvironment, right? But if we have a process that regulates phagosome maturation, that has components that are exclusive of this process and has nothing to do with the subsequent maturation of the phagosome or the autophagy pathway, and then I'm talking specifically about the lab components, I think that these are great candidates, even though we don't have any strict drug designed for that yet. So you can mess up with the process of, mess up, it's a terrible word. You mess up, you modulate the process that it's contributing to immune suppressive response, immune suppressive activation in response to engulfment of dying cells in a specific way without messing up with the cell physiology. So I think that they are potentially very nice candidates, but everything is brand new we don't have like any, I'm sure a lot of people are working on that.
So Larry, you said that phagocytosis links to autophagy machinery in dying cells, in infection, particles, zymosan, etc. Is there a situation that we don't have LAP associated with phagocytosis?
Yes.
And also, if it's always the same, it's always autophagy, why? In which point? Do you think it's in the recognition or in the later phase that cells that engulf apoptotic cells respond in a way different than the cells that are engulfing pathogens?
I think that down to the point where you have LC3 lipidation to the phagosome membrane, because of the phenotypes between dying cells, zymosan, PAM3 covered beads, they are so similar. I think that I would expect the components to be the same. The secret for the different responses, I think that it's gonna be in the sensing of the cargo. And by saying that, I don't want to say that necessarily what's being destroyed in the phagolysosome. It might be that you have a way to recognize if what's going to be degraded. Alguém pode me corrigir? É degraded or degraded? I was confused.
Doesn't matter.
Yeah.
It matters.
Degraded.
Thank you, degraded. If the cargo being degraded, it's a dying cell or it's a bacteria, right? But in terms of what it's the connection between the different receptors and the triggering of LAP, I think this is the Saint Graal of LAP right now, right? What's going on there at the beginning of the process? We know that Rubicon, it's recruited very early on in the process before you have phagosome closure. But what's upstream of Rubicon?
All right. Larissa, I think we have other questions showing up, but I think we should move on for now, and we keep the last questions for then, because we have just an hour, half an hour.
No, it's going to be enough. No, don't worry. So all that I talked to you right now, it's kind of connected with this idea that we established of a direct relationship between the type of cell death and triggering an effector function. That apoptosis is going to be immunosuppressive, pyroptosis, it's going to be immunogenic, necroptosis is going to be immunogenic, either if it depends on engulfment or just because you're going to have damps in the circulation. But I really like this picture. This actually is freely inspired by a very nice review. It's not only a review, it's more a perspective by Carla Brothley, published last year. And I highly recommend to the students reading this paper. It's a very nice work, where she proposes, and I think this is aligned with the kind of data we have right now, that you have actually an integrated relationship between several factors that are occurring during efferocytosis. So to understand what's going on in terms of what kind of effector functions being activated in response to efferocytosis, you need to get into consideration definitely the type of cell death, and this is obvious if it's going to be an apoptotic, necroptotic, pyroptotic, because this is going to define the eat-me signals that are being exposed, or the timing when they are going to be exposed. If there is damp release, if there is associated inflammasome activation and the consequences of that, if there is complement activation and the consequences of that, if you're having release of NETs or other traps that can be induced in response to pyroptotic, at least, cell death. And this is the most well accepted concept. But you also need to take into consideration the cell identity. The cell type matters. Even though phosphatidylserine is an universal trigger for efferocytosis, it might be that phosphatidylserine in an epithelial cell, it's not interpreted as phosphatidylserine in a macrophage, okay? And obviously, if it's a sterile kind of cell death, or if the cell is infected. You also need to take into consideration the nature of the efferocyte, the genetic programming of this cell, and what this cell is actually capable to do, how it deals with cargo. So we don't know if all the cells are going to respond to engulfed dead cell in the same way, and the metabolic changes that occur in this efferocyte in response to engulfment of a dying cell. And you definitely have a role for the environmental input. So while you're having efferocytosis, what's going on around the professional phagocyte? What is the cytokine milieu? What is the kind of, what is the tissue? Who are the components in this microenvironment? What is the extension of the injury? Because efferocytosis is bona fide mechanism to promote tissue repair. And the amount of trash that we are talking about is going to matter. So I think that we should keep in mind this integrated relationship between death and clearance of death. I think it's way more complex that we've been interpreting it. And from this point on, I'm going to talk a little bit about lab and this activation of nucleic acid sensing. As Gustavo mentioned, I think this is fundamental. But other than that, I'm going to show some literature, but not dig in. I think that these are just things that you should keep in mind. And I hope it's just helping to draw this big picture. And I'll talk first about, so I'm not going to talk about the specifics of the type of cell death. Again, you're going to have a lecture on that later this week about immunosuppressive versus immunogenic. I don't think I can focus on the other points. And as Karina and I were discussing a little bit earlier, phosphatidylinositol externalization is not exclusive of apoptotic cell death pathway. And even though I'm not going to talk about the specific types of cell death, I just want to put this flag here to keep in mind. Phosphatidylinositol is present in a cell that it's dying by apoptosis, and this the presence of phosphatidylinositol on the outer membrane happens early on, but this is going to happen in response to pyroptotic cell death, and this is going to happen, this is going to happen in response to necroptotic cell death, and this is going to happen in response to pyroptotic cell death, with completely different timings. But still, that means that sensing of phosphatidylinositol to trigger efferocytosis can occur in response to any kind of dead cell, potentially, but that's going to happen in the context of sensing DAMPs and inflammatory cytokines that are coming from this other immunogenic cell death. And these should somehow affect both how a professional phagocyte engulf a dying cell and how this engulfed corpse is going to be interpreted. I don't think this is a simple relationship. But let's focus then in the environmental input and in the characteristics of the efferocyte that are going to contribute to the outcome of efferocytosis. And in terms of the environmental input, even though I put like right in the beginning of the presentation, we have this idea that dying cells and Th2 cytokines, they obviously are going to drive anti-inflammatory and pro-resolution responses. But it wasn't until like 4 years ago that this has actually been experimentally proven in a very elegant paper, actually. So if a macrophage is going to drive a full tissue repair program, that requires both engulfment of an apoptotic cell and the presence of Th2 cytokines, at least in these scenarios where you have induction of Th2 cytokines. So either of them, they cause weaker phenotypes in this macrophage, which tells us that actually efferocytosis can act as a signal one for a full-blown tissue repair programming of macrophages. And if you impair efferocytosis, and here by treating the dying cells with annexin-5, which is going to block the recognition of phosphatidylserine because it binds to phosphatidylserine and now it cannot be recognized by the efferocytic receptors, then you see reduction of the expression of genes that are related to tissue repair programming of macrophages. So it seems straightforward, but it wasn't proven until very recent. And in terms of the characteristics of the efferocyte, phagosome maturation matters. If phagosome maturation doesn't proceed, if you have defects in the pathway, and this can be represented by defects in lab, or if digestion of the genetic material of the engulfed dying cell doesn't occur properly, you can have activation of immune responses. So this is in vitro experiment. These are macrophages treated in a plate. So if you transfect DNA, you're going to have production, you're going to have stimulation of interferon, type 1 interferon expression, and this doesn't require a lab. But now, if you feed a dying cell to these macrophages, and this little bar here is the wild type cells, you don't see activation of type 1 interferon, and this is expected because if you have, if efferocytosis proceeds as expected, you're going to have degradation of the genetic material of these engulfed corpse. But if you have a defect in this pathway of phagosome maturation, now you start to see production of type 1 interferon in response to the dying cell. And one of the sensing hubs for the presence of foreign DNA or out of place DNA inside a cell, if you have DNA in the cytosol, is STING, and this is a major adapter for the production of type 1 interferon. And this production of type 1 interferon in response to failed maturation of the phagosome is actually dependent on the activation of STING, which tells us, and this might be an oversimplification, it might be for another reason, but there is a very strong possibility that if you have a fail, if you have a fail in phagosome maturation, if this corpse is not efficiently cleared, you might have nucleic acids, you might have DNA that actually leaks to the cytosol, and this is sensed by molecular platforms that trigger type 1 interferon production, and these can be linked to autoimmune, but this can also provide protection against tumor growth. And then we can experimentally prove that. So if effective efferocytosis in some scenarios is immune silence, and we know that this is happening in the tumor microenvironment, and focus first here in the first graph, you have, if you eliminate the machinery for efficient clearance of dying cells, you have slower tumor growth, and this is caused, and I'm not showing the data, you need to believe me, this is caused by activation of immune response. Now you have these macrophages become inflammatory in response to dying cells. And this depends on activation of these STING platforms in response to DNAs, to the presence of, possibly by the presence of DNA in the cytosol, because if you eliminate STING, now the tumors go back to growing, okay? And I'm gonna show you some other examples of how the programming of the efferocyte matters to the outcome of the response. And the next couple of slides are actually about metabolic links between efferocytosis and degradation of dying cells, and the induction of anti-inflammatory and pro-resolution or pro-tissue repair programs. So we now know that once this corpse is degraded, you have active release of liberation of arginine in the cytosol, and arginine processing pathway that it's triggered by arginase 1. And here you have a whole signaling pathway that I'm not going to talk about. You can, this is a very nice paper, I super recommend. But in the end, what happens is that you're going to have activation of actin polymerization machinery, and this is going to be important for continual uptake of dying cells. And if you don't have continual uptake of dying cells, you don't have optimal tissue repair, which means that defects in this pathway of the efferocytes that has nothing to do with the cargo is going to impair proper tissue repair response to efferocytosis. Another very recent and very elegant work, and this is actually the first demonstration of, to the best of my knowledge, where you have lipid products of the degradation of a dying cell being directly recognized by machineries, molecular machineries from the efferocyte. So fatty acid released from cell corpses by proper clearance. This is going to activate fatty acid oxidation pathway and mitochondrial respiration, and through also a very elegant, a very complex and elegant molecular pathway, you're going to have activation of this transcription factor, PBX1, that binds to the promoter of IL-10 and trigger IL-10 production. And this mechanism is important to proper tissue repair in a model of cardiac infarct. Okay, which means that metabolites from the engulfed cells, they can dictate how the macrophages are going to optimally deal with several dying cells in the environment, but that requires signaling from the professional phagocyte, not only the elimination of the dying cell per se. And the last point that I want to talk about is the fact it's whether the cell is sterile or infected. So cell identity is not only that, as I mentioned, the cell type, what tissue is coming from, can affect how efferocytosis, what's going to be the outcome of efferocytosis. But I think that the most obvious scenario is when you have a sterile cell being engulfed or an infected cell being engulfed. So there's a lot of nice literature. I just selected a few papers to show that a sterile corpse and a sterile apoptotic cell is not sensed by the macrophage the same way that an infected apoptotic cell. So you have the same type of cell death, of programmed cell death, but the outcome is going to be different. And that happens because the macrophage can sense, can discriminate a sterile dying cell from an infected dying cell. So this is beautiful work showing that engulfment of infected dying cells, cells infected with E. coli or with Citrobacter, they are the relevant physiological inputs to trigger synergic production of TGF-beta and IL-6. So the combination of TGF-beta and IL-6 in vitro can drive Th17 differentiation. But for a while, it wasn't known what is the source, the physiological source of the combination of these two potentially antagonistic cytokines. And one source of these cytokines is the engulfment of infected apoptotic cells. That's going to trigger production of TGF-beta and IL-6 at the same time. And this is going to be important to skew the differentiation from Tregs to Th17 cells. And not only that, but later work actually from Alexandra Medeiros here from Brazil showed that besides TGF-beta and IL-6, efferocytosis of apoptotic infected cells, infected with either E. coli or Citrobacter, is also gonna induce the secretion of prostaglandin 2. And this is also acting on the regulation of this TH17 differentiation, regulating regulating in a negative way that can compromise adaptive immunity in the end. So you have a very complex relationship between recognition of infected apoptotic cells and the outcome in terms of the immune response. This is another story that I think it's really nice, showing that efferocytosis of apoptotic cells, if they are infected with a bug, and the bug here in question is Mycobacterium, this can be important to drive antibactericidal mechanisms. So antibactericidal mechanisms are a bona fide inflammatory phenotype of macrophages, and usually it comes in response to recognition of a dying cell, of a pathogen, plus a Th1 cytokine. Clearance of TB, TB actively impairs its own clearance, but if a macrophage engulfs an infected cell, now you have proper activation of antibactericidal macrophage mechanisms to eliminate these bugs. And finally, I'm going to talk about a story from the lab. So we've been working on COVID-19. I'm going to show data from that we think it's a connection between efferocytosis and the immune deregulation that occurs during COVID-19. So I think that by now everyone, it's well accepted, and it has been shown by different groups that severe COVID-19 evolution from a moderate case to a severe case is actually connected to deregulated host responses. So not necessarily to a failure in the clearance of pathogens, but to immune response that got so bad that you have like a system crash because physiological balance is completely lost. And because one of the signatures of this deregulation of the immune response, it's both hyperinflammation and extensive tissue damage, we thought that maybe efferocytosis could be a player driving this immune pathology during severe COVID-19. So just a few slides showing you some data. Actually, we first noticed that dying cells, they carry viable SARS-CoV-2. So if you label dying apoptotic cells, and I'm not showing the data of the apoptosis, but infection of epithelial cells with SARS-CoV-2 drives apoptosis. But here we are marking one, the spike protein from the virus, and we see that these apoptotic bodies, they are spike positive, right? So spike is in the surface of the virus and indicates for us that you have viable virus inside these corpses. But we tested that experimentally, and if you purify these particles from the dying cells, then you have viral infection, showing that they are there and they are viable. And these infected corpses, they are taken by macrophages, at least in vitro, right? So here it's just an assay, it's just a representation of a dying cell where we also label this spike protein, and this cell can be actively engulfed by the macrophage. Here is just a simple quantification. But here's when things starts to get interesting. So we noticed that engulfment of infected apoptotic cells cells infected with apoptotic cells infected with SARS-CoV-2 skews or shifts the response to efferocytosis. So going over all this colorful thing, it's the same scheme over and over. So this is just our control untreated cell. This is a cell treated with an UV radiated apoptotic cell. This is a cell, apoptotic cell that has been infected with CoV-2, and this is simply the supernatant of the infected dying cell. So this is going to contain released viral particles. It's going to contain possible damps from the dying cell, cytokines released by the dying cell, okay? And we are also giving that to the macrophage. So this is what we expect in response to an apoptotic sterile cell type. You see upregulation of CD206. This is a classic marker of anti-inflammatory commitment of the macrophage. This doesn't happen if the cell is infected with CoV-2, but on the other hand, the stimulation of macrophages with these infected corpses triggers the production of inflammatory cytokines. Here I'm showing IL-6, IL-1 beta. And this is a response that it's being triggered by the viable virus inside the apoptotic cell. And how do we know that? If we take these infected corpses and we inhibit viral viability by UV radiating them on top of infected them, or if we fix these corpses with formaldehyde, then we have a reversion, and this is important, of the CD206 expression in the surface of the macrophage, meaning that it's not that the virus modified the type of cell death that now it cannot be anti-inflammatory. The virus inside the corpse encodes information that shifts the interpretation from anti-inflammatory to inflammatory. And if you eliminate viral viability, now the macrophage can interpret this body as an anti-inflammatory signal. And now you don't see production of inflammatory cytokines anymore. And here's what becomes more interesting, at least in my point of view. We checked the expression of several of these eat me receptors, scavenger receptors, TIM4, TEM receptors. And we consistently notice, and please pay attention here to the light blue bars right in the middle. For most of them, but not all of them, see the 10 receptor Merk. You don't see difference, and this is important because I'll talk about it in the last slide. But most of them, they are downregulated in response to efferocytosis of an infected body. The same happens when you directly infect the macrophages with SARS-CoV-2. But then again, it is a promiscuous relationship. It doesn't happen to all the receptors, okay? So you have an effect of both SARS-CoV-2 and the uptake of SARS-CoV-2 infected dying cells in the expression of the receptor, of the efferocytic receptors. And this seems to be very important, at least in our model. So here we tested if this observed reduction in the expression of efferocytic receptor mattered. And for that, we tested if macrophages that have seen an infected an apoptotic cell infected with SARS-CoV-2, if they are able to clear other apoptotic cells. And just remember from everything I told you, the capacity to engulf several cells in continual cycles, it's fundamental for optimal tissue repair, at least in an infection scenario. So if a macrophage has engulfed an apoptotic cell carrying SARS-CoV-2, then they become blind to other dying cells. So they are not able anymore to clear apoptotic cells. We also see the same thing if the macrophages are infected with CoV-2, but definitely not to the same extent. So you have a massive reduction here in response to an infected body, but not the same thing in response to direct infection with CoV-2. meaning that engulfment of SARS-CoV-2 infected apoptotic cells suppress the capacity of macrophages to continually engulf other dying cells, and this might be important for impaired tissue repair during COVID-19. And to finalize, we can see in the lung of patients that died of COVID-19, a post-mortem autopsy, that there is a reduction in the expression of this efferocytic receptor in infiltrating monocytes and monocyte-derived macrophage in the lungs of patients with COVID-19. So here we are looking to CD36, the same with MERK, the one that we don't see difference in the gene expression, but we can see difference in the expression in the lungs of the patients. So this is what we think it's happening. So when you have the engulfment of an sterile apoptotic cell, as I talked to you, this is the bona fide efferocytic stimuli that is better characterized. You have activation of anti-inflammatory and tissue repair programming. But if this apoptotic cell, and it has nothing to do with the nature of the cell death, but the fact that it's carrying SARS-CoV-2 with some sort of Trojan horse mechanism, this is going to have at least two important consequences in vitro, that it's the production of inflammatory cytokines and the reduction in the expression of surface expression of efferocytic receptors and impaired uptake of other apoptotic cells. And both of these, and these two phenotypes might contribute to the hyperinflammation and spread tissue damage, and that it's associated with severe COVID-19. So the take home here from this, 1 hour and a half that I talked is uptake and processing of dying cells by innate immune cells regulate inflammatory responses, not only of macrophages, forget this last piece. But I think that the most important message here is that the outcome of this process may change according to the dying cell, to the efferocytic cell, and to the environmental input, and it's not straightforward as we would like to think. And with that, I'll finish. For this last bit, there is a lot of people in this work that I would like to acknowledge very quickly. And hopefully there's time for a couple of questions. Thank you very much, guys.
Thank you, Larissa. It was an amazing talk, a lot of things to think about and discuss. It's a pity that we cannot go for lunch all together and have long conversations. But for example, I'm dying to know if your model there depends on STING receptor or not. But I'll ask the questions from the chat here. Mm-hmm. The first one here is about polarization of macrophages in this situation. Of
polarization?
Yeah. So does this process of LAP engulfing cells and processing it in different ways help polarizing the cells in a way or the other? And also, if a cell is already polarized, the macrophage or the disease, does it interfere in the LAP ability of the cells?
Okay, to the first question. So LAP of sterile apoptotic cell drives anti-inflammatory programming. Yes. What we've been studying, and it's very preliminary, but I think I've seen enough data, is that if you have a failing LAP and you activate STING, it's not STING that it's inhibiting the anti-inflammatory programming. So I think you have different sets of pathways being activated. That's the message. I've never performed I've never polarized the macro, even though it's obvious, but I never polarized macrophages and then test the efferocytic capacity. I'm sure this has been done. I know that you can shift the programming after, but I'm sure there is data on the other direction. I'm just not remembering right now. And I've never done those experiments.
Okay, Karina, can you follow up?
The last question here. So, are there any advantages of LAP over phagocytosis in terms of pathogen clearance and perhaps a specific trigger for LC3 recruitment?
Yes, there's been quite a few interesting papers in the last couple of years showing how LEP is both modulates and it's modulated by pathogens. So it does. So there is a very nice experiment with one species of Legionella. I forgot the name. That they infect the macrophage. So some Legionella are driven to LEP, others are not. So it seems that you have some sort of regulation on this response, but I don't think it's clear how.
So Lari, thank you very much for this
amazing
talk.
Can I just, sorry, I meant to interrupt you when you talk about recognition of necrotic cells also via phosphatidylserine. I just want to mention, particularly to the students, that besides different kinetics of phosphatidyl externalization, as you mentioned, we have two other situations, I wouldn't say problem, when we follow up phosphatidylserine in those set of experiments. First of all, is that the externalization of phosphatidylserine by necrotic cells are more randomly, are not coordinated by caspases, for instance. So you have much less externalization or residues in the outer leaflets compared to apoptotic cells. And secondly, from those experiments, you cannot really tell if the phosphatidylserine residues are inside or outside the cells, because at the time that you show and people look at you can see blebs. So you see that the membrane are already permeabilized.
Yeah.
And annexin 5 just go inside the cells and label the inner leaflet. So you have both situations, and I think if people want to address those questions in their experimental setups, then they need to try to block or do some other complementary experiments.
Yeah, I think the classic, so this in vitro experiment where you incubate dying cells with annexin 5, it's a straightforward way to test this, because taking one on another receptor is not gonna, you might see a difference or not, but.
Yeah, you can use unlabeled annexin 5
unlabeled, yeah.
to block the interaction with phosphatidylserine or stuff like that.
I have just to point out as well, Gustavo, that at least in necroptosis, and we might see it tomorrow, there is a very early externalization of phosphatidylserine, much earlier than the permeabilization of the cell. So it seems that it's a lower level, as you said, but it seems that there is a concerted effort of the cell, or at least a mechanism that externalizes phosphatidylserine in these settings. So it's not only about like making holes and staining from the inner leaflet, but it seems that in necroptosis there is a flipping to the outside leaflet as well.
Yeah,
but that's
what I said.
Yeah, there's a huge field there to understand how is this regulated, right? Gas.
I'm sure that these experiments are being done somewhere right now.
Yeah. So Larissa, thank you very much. I'm sorry that we have to interrupt the discussion, but just reminding everyone that tomorrow is the same time, same place, same link. So if you have trouble accessing, just close your software and your tabs and open it again. Just insist a little bit that you'll be able to connect. All right. Again, thanks, guys.
All right, everyone.
Here.
Thank you so much.
Bye.